Welcome back. This is part 2 of the Chapter 15 AP Biology lecture. We left off in the last video talking about X linkage. Now let's look at Y linkage a little bit. Hairy ear pinnae, kind of like having hair in your ears, is thought to be Y-linked. There are tons of new techniques that have found a number of Y-linked markers that seem to be running in the males of a family. So that's sort of an emerging science. There's some emerging data. Your textbook has some good data on those Y-linked traits. Sex-influenced traits are those traits that differ because of the hormones of the sex, things like beards and mammary gland developments and baldness, not male pattern baldness, but different types of baldness. These are not on the sex chromosome, but they're a result of, they're sex-influenced. They're not sex-linked, they're sex-influenced. There's baldness. Male pattern baldness is an example of that. Males, if you have the gene, you're bald. Females, if you're homozygous, then you have thin hair, or you have that male pattern baldness pattern. You see that sometimes in females. There are females on those male pattern baldness with Bosley commercials and those types of things. So a bar body is an inactive X chromosome observed in the nucleus. So that's one way of determining the genetic sex without doing a karyotype. So basically, females have two X's, right? So one of those X's becomes deactivated. Which one appears to be random? Now, you know as well as I do, often in science, something appears to be random until we collect more data. But as of now, it appears to be random. So that inactivation happens early in embryonic development. We add methyl groups to the DNA. So you'll see that described as methylation. We add a methyl group, we're adding a different functional group to the edge of the chromatin, and that changes the electronegativity of the region. That's going to change the folding of the chromatin, and it's going to change which X is activated and which is not. Incidentally, this is not unique to the X chromosome. We methylate lots of our DNA. So the result of this X inactivation is that body cells tend to be a mosaic of the X types. Calico cats are an example of this. We have some examples like sweat gland disorders where some work, some don't. In the case of calico, black fur is XB, XO is orange, and then calico is the heterozygous XB, XO. So I mentioned it's kind of like codominance, but rather than codominance, we're inactivating one or the other. So here in this picture you see the active X is the orange allele, and in this case the active X is the black allele. So it's not dominant recessive, it's not truly codominant, it's an inactive X. There's a bar body there. Check that out. Why don't you find many calico males? Because to be calico, they have to be XBXO, Y, to be a male. So XBXO makes calico, Y makes them a male. So that means they have 3 sex chromosomes, so they're sterile. They can't reproduce. So typically, most calico cats are female, just like most, never mind. Okay, so chromosomal alterations, moving on. How can chromosomes change? We know that mutations are changes in genetic structure. How can chromosomes mutate? Well, we can change the number or we can change the structure. In terms of number alterations, we're talking about aneuploidy and polyploidy. Aneuploidy you've experienced with Down syndrome, trisomy 21, trisomy 13, those types of things. Polyploidy is going to be a whole set of chromosomes. So where we have 46 chromosomes, they double, and now we have 92 chromosomes in an organism. You can imagine that's a pretty drastic thing that doesn't happen very often. I'm going to run through this quickly because we covered it in meiosis in semester 1. Great chance to recap that, though. A non-disjunction is going to be when chromosomes don't separate during meiosis. So take a look at this picture. Non-disjunction happens in meiosis 1. We end up with 4 gametes that all have an altered number of chromosomes. 2 of them have too many, 2 of them have not enough. If our non-disjunction happens in meiosis 2, this offshoot isn't affected by the mutation. This one is. So if someone were to ask you on a test or something like that, which is quote-unquote better, a non-disjunction in meiosis II is going to be better. So there's still a 50% chance of having an offspring that has the correct number of chromosomes. There are two types. We could have a monosomy where that's where we're one short. We could have a trisomy where we've got one extra. Turner syndrome is an example of a monosomy. See, the genotype is X blank or X0, depending on which type of notation you use. The phenotype is female, but they have poor secondary sexual development. Sometimes they're infertile. Not always, but sometimes they are. You see some of the characteristics there of someone that has Turner syndrome. So why are they usually sterile? Because they have an odd chromosome number. The extra X chromosome is important for ovary development. So things just don't match up right, and when things don't match up right, the body tends to shut down fertilization events. There are some other sex chromosomal changes. Maybe you're looking at one of these three as part of your inquiry, maybe not. Oops, sorry, skipped. Trisomy events. Trisomy 13 is sometimes called Patel syndrome. Trisomy 21 is called Down syndrome. That's where we've got an extra chromosome. You see here in the karyotype 21, there's an extra. Now notice that trisomy 21 and trisomy 13, which is here, are the two most common, not trisomy 1 or 2. Trisomy 1 and 2, well, those chromosomes are really, really long. So an extra one of these is a lot of extra genetic material, whereas an extra 21 is just a little bit of extra DNA. So typically in nature, the smaller the change, the better chance you have to survive, typically. So why is a trisomy more common than monosomy? Same kind of idea. Fetus can survive an extra copy, but not having enough is usually fatal. In terms of a polyploid, that's where we've got triploid and tetraploids. Usually that's going to be fatal in animals. For some reason, plants, particularly trees, tend to be more flexible. They can handle polyploid events more frequently. But in those plants, even number of polyploids are often fertile and odds are not. So you can imagine that if we have a polyploid event where we double our DNA, that's going to be a huge evolutionary split in a species. We're probably looking at a speciation event there. Think about the fact that plants can self-fertilize. So if there's an issue in meiosis, there's a good chance that there are going to be multiple issues in meiosis, and you're self-fertilizing, so your DNA could potentially double. Again, odd numbers are weird when it comes to reproduction. The organism tends to recognize that something is amiss, and we should do something else, or we should terminate the fertilization. The chromosome can structurally change too. Most of these, we can look at them and be like, oh yeah, that makes sense. Let's look at the picture. So deletion. We deleted part of a chromosome. That makes sense. That's going to change it. Instead of being ABCDE, we're ABCE. We could duplicate a segment where we have two BCs here now. So now we've got a longer chromosome. We've got extra genetic material. We could have an inversion where they flip. Instead of ABCDE, we have ADCBE. Again, that's going to change the structure. That's going to change the expression. It's going to change the chemical structure. It's going to change the folding pattern. Translocation is going to be moving from one place to another. So maybe we're talking about crossing over here. Typically, we're talking about crossing over. But we're just swapping information from one chromosome to another. There you see some chromosomes that have been lit up because they've been altered. So what's the result? We could lose some genetic information. We could get some extra genetic information. Or we could have DNA in a place that is not expected to be. Often, we haven't gotten this far, but often one gene's expression is influenced by its location to other genes. There are promoters and there are silencers and things like that that can manipulate our expression. And if the gene moves to a new address, the promoter can't find it. So effects can be drastic or can be smaller. They can be terminal, they could be fatal or not. Cri du chat is an example of one where we tend to see a lot of survivors of Cri du chat syndrome. Part of the P arm of chromosome number 5 is missing, so there's been a deletion. Again, they survive, but there's low birth weight, and they tend to be small. It's called Cri du chat because they sound like a cat when they cry. That's French for cry of the cat. And they tend to be severely mentally impaired, and they have small heads. So there's a picture of a young lady with Cri du chat syndrome. Something called a Philadelphia chromosome. You can imagine where that was discovered as an abnormal chromosome with a specific crossing over between 9 and 22, chromosomes 9 and 22, which causes that myeloid leukemia, which again tends to be pretty fatal. There is something called parental imprinting of genes, where gene expression and inheritance depends on which parent passed on the gene. Think about that from the Mendel discussion. We said that was one of the spermists and the ovists. That was one of the ideas that Mendel kind of redirected. Usually this is caused by different methylations of the DNA, different processing. So something gets a male methylation pattern or a female methylation pattern, and that makes you more like your parent. Crazy enough, it resets every generation. So this kind of shows the maternal imprint on the chromosome in the egg, the parental imprint on the sperm. They meet up. There's inherited imprinting that impacts the development, sort of those coordinated processes, and are you going to be tall like your dad, short like your mom, those types of things. And then, so this generation continues. This generation grows up, and their original imprints get erased in their sperm and eggs in the cells that create those. So there's a new set of imprinting between one generation and the next. So so the imprinting that I received from my dad and that I received from my mom gets reset. So I don't pass on my dad's imprinting and my mom's imprinting. I pass on my imprinting. So that's where the whole nature versus nurture thing pops up in genetics, because the way that I live my lifestyle is going to impact the imprints that I make and that I pass on to my children. Quickly want to run through an example here, a couple of examples where a small gene region from chromosome 15 can, if it is in a male, you get Prader-Willi syndrome, and if you get the female imprint, you get Angelman syndrome. So that's not saying that all boys born with this are going to have Prader-Willi and all girls are going to have Angelman. It has to do with the imprint that is most dominant. So if I received that male imprint from, or if I received that lack of a chromosome 15 from my dad, I would have this. If I received it from my mom, I would have this. Slightly different, but definitely different. So those imprints are erased in those gamete-producing cells and recoded by my body as male. Even if I received it from my mom, it gets recoded in my body as male. So phenotypes don't often follow or don't always follow Mendelian inheritance because the sex of the parent does matter. Quickly, there are some things that are inherited extranuclearly that don't follow this pattern. Mitochondria and chloroplasts, because of that endosymbiont theory, we know about that. That has nothing to do with crossing over or chromosomes or things like that. Again, Mendelian inheritance patterns fail, especially where traits are passed from the egg, and that's mitochondria, chloroplast, same idea. So there's variegation in plants. You can see that there are different color patterns due to imprinting and the chloroplast DNA being passed from one generation to the next. I don't want to rush too quickly through mitochondrial. That's okay. Epilepsy, red fiber disease, these are all things that are associated with ATP generation. So that's where the mitochondrial DNA can produce issues from one generation from the next. Shouldn't be surprising that they're related with ATP generation. We can have a few small organelles and we can have some healthy ones. So it depends on how much of the mutation you inherit from your mom. So that's the chapter 15 lecture. Let me know if you have questions.